Hello, and welcome to the Gaming with Science podcast, where we talk about the science behind some of your favorite games.
Today, we will be talking about Diatoms by Ludoliminal Games. All right, everyone, welcome back to Gaming with Science. This is Jason.
This is Brian.
And today, for our special guest, we have Laura Aycock. Laura, can you please introduce yourself?
Sure. I'm Laura Aycock. I am the collection manager of the Diatom herbarium at the Academy of Natural Sciences in Philadelphia that's affiliated with Drexel University. And I've been working with diatoms for about 15 years, and I find them fun and enjoyable.
That's really cool. Thank you for coming on. Jason, how did you manage to get the exact right person to come talk to us? Good job.
Being very persistent with emails.
It's also not very many of us.
There is that. When there's actually a website called diatoms.org that has all the nation's top diatom scientists linked to it somehow, it's not that hard to find someone. So before we get into this lovely game about absolutely beautiful microscopic creatures, let's start with our fun science facts. So Laura, as our guest, we usually pass the privilege to you to start. Do you have something you'd like to share with our audience?
Sure. My favorite fact about diatoms is they produce about a fourth of the oxygen we breathe. So they're very important to life on Earth, and we wouldn't survive without them.
So trees get all the credit, but they're stealing that.
We talk about plant blindness, where people just don't look at plants. There's definitely macroscopic bias, where we just don't think about all the things that aren't within, human size scale. So yeah, trees get all the credit, but all these little microbes are actually doing a whole bunch of the work there.
Yeah, diatoms, along with other groups of algae, actually produce about half of the oxygen we breathe. So they are as important, if not more important, than land plants. But no one thinks about them, sees them, or really acknowledges them.
So let me think then. I'm thinking about this track of carbon dioxide that we've been seeing sort of dip and rise and dip and rise and dip and rise. Now that dip and rise, that's from the alpine forests in the northern continents, right? But the stable activity, that's presumably all the algae in the ocean, right? Or do they also fluctuate on an annual cycle?
I'd assume they'd also fluctuate annually just because of temperature, if nothing else.
It depends on the environment. So diatoms in the ocean are relatively consistent, but I think it does fluctuate with temperature. I actually don't know too much about marine diatoms because my expertise lies in benthic freshwater diatoms.
Benthic freshwater. So that means the things that live in the muck at the bottom of freshwater environments?
Yeah, the brown slime you see when you go to creeks. That's what I love to look at.
Oh, you're a slimeologist. That's awesome.
All right, Brian, your turn. What fun fact do you have for us today?
Well, funny enough, I also brought a diatoms one. I was looking for something recently about diatoms in the news. It's a press release out of Stanford about diatoms remaining active down to negative 15 degrees centigrade. So cold. Basically, in solid ice isn't as solid as you'd think. Actually, it can have these little microfluidic chambers within it, sort of threads of liquid water. And the diatoms were actually not only colonizing these, but moving through these chambers. So I didn't even know diatoms could move. I guess they have little actin filaments that they use to move on slime. I want to know more about this, and I'm hoping that Laura can explain it.
Diatoms are very capable of active movement. Not all of them, though. They have to have a slit in the center of the cell, which is called the raphe, and they can secrete mucilage, and they glide along like slugs.
So you can tell just by looking at them if they're going to be able to be mobile?
Yep.
Do all the ones with raphe have mobility, or do some of them have the raphe and are not modal?
No, all of them have mobility. The raphe can vary in its placement on the cell, whether it's in the center of the cell, along the sides, if it's on one half of the valve, because diatoms are made in two parts. They're kind of like a box where you have a top half and a bottom half. So when they're dead, they split apart. So you'll see the raphe on one valve and not the other, but they do have the raphe.
That's really cool. Man, so diatoms are kind of like mimics in D&D. They live in a box.
They are a box.
A glass box they make themselves.
That's okay. These are very cool organisms.
They are. So let's go on to this game then, because this game is a beautiful game about these beautiful creatures. So Diatoms is a game by Ludoliminal Games and published by 25th Century Games. It actually won a 2025 Mensa Select Award. And I like the tagline on the publisher's website. It is a stunningly beautiful game about making art from algae, which is not something you would think about. But the whole metaphor of this game is about Victorian diatom art, which is this obscure art form where back when microscopes for the masses were a newfangled thing and people were trying to sell them. They wanted to sell things that you could look at right away. And so they would sell these little slides you could put under, and they had diatom art on them, which is what you're making in this game. We'll talk more about what diatom art is in a little bit. For the game itself, it's basic stats. It's for 1 to 4 players, obligatory single player mode. Although I'll say this is one of the few games we've played where I actually have played the single player mode, and I can attest it's actually quite fun.
Yeah, I was going to say, you actually said you liked it. You enjoyed it.
It's very calming. And ages 8+, about 30 to 45 minutes to play. Suggested retail price price is $55. A lot of that is probably going to the very high quality components. So there's very high quality chipboard, most of which has foil embossing on it in some degree, oftentimes lots. The game is played in two sections. You have your tile placing one, where you've got these hexagonal tiles that have colors coming off of them. So every hexagon consists of 6 triangles joined at the tip. And so those 6 triangles can be any one of a number of colors. They've got 5 different ones: red, yellow, green, blue, and purple. Some of them are white as wild spots. And it's a typical like color matching game. You have the hexes down on the board, and then you try to place new hexes so that the colors match. That part is fairly straightforward. The thing is, based on the colors you make at that intersection, so when you place a hex down next to 2 other tiles, it forms a point where all 3 of those tiles touch, and where therefore there are 6 triangles around that central point. And the size and distribution of the color patches determines which diatoms you then collect. Metaphorically, this is you like looking at a patch of water under the microscope and like sucking it up and being able to grab some diatoms out of it. The second half of the game, every player has their own little player board. So everyone's working individually here. You've got the shared water tiles, but an individual board. And you are placing those diatoms on the board in order to make arrangements. And the board has all these cutouts where I haven't counted the number of spaces. There's probably like 40 or 50 of them. Each one of them can only hold one of 2 shapes. And they've got it drawn out. So you can say, oh, this spot can either hold a triangle or a star, but it cannot hold a circle. Or this one can hold an oval or a circle, but it can't hold a square. That sort of thing. Because the diatoms, in addition to coming in 5 colors, also come in 5 shapes. And those shapes are based on the size of the color patch you made when placing those tiles together. The idea is you're trying to place these down artfully. And of course, because it's a game, it's not just aesthetically pleasing. They have rules for how you gain points. And when I have taught this game to other people, which we did at a few conventions last year, keeping track of the scoring rules is the hardest part of this game. People get the tile laying pretty easily. People get getting the shapes and collecting the diatoms. Remembering the many, many different ways you can score points is actually the hardest part, because you can score points based off of how many of different colors you have. More diatoms of a single color gets you more points. You can get them based off of where they are. So the board is circular and it has kind of 3 rings. So you have your inner ring, your middle ring, and your outer ring. And then based on the diversity of shapes you have in those determines points there. And then there's all these lines of symmetry. So horizontal and vertical and diagonals. And if you have matching pairs of diatoms on those on the same ring, the score sheet for this is thankfully very well put together. There are literally like 20 or 30 spots for you to write down, oh, here's how many points I got for this particular arrangement here. And then this one here. It walks you through. If you just go through the score sheet, you will have everything, and it makes tallying it up at the end easy. It does not make keeping all those rules in your head during play any easier. That is still quite hard. And I think a lot of people, at least a lot of people I've played with, reach the point where they focus on a few things and they just kind of don't worry about the rest. Even after several plays, when I'm looking at this, like, okay, I can keep a few things in head, but I can't keep all possible ones in head. So sometimes it's just like, well, I'm just going to play this here, get a few things, and then I'll figure out where I can place them well.
I think that's everything in the game. When you're talking about components, one of the things they have are these little petri dishes that you get to keep all your pieces in. Except I can tell you, if you've ever worked with actual petri dishes, these are much nicer than regular petri dishes. These are actually like your normal petri dish. If you handle it wrong, it immediately cracks. These are actually meant to hold up over time.
Yes, these are not meant to be disposed of after a few days.
No, for sure.
And they have a few other things. So they have like every player has their own little like score guide book so that you can look through and you can see how the scoring works. They've got this cute little magnifying glass so you can like isolate the little 6-point section that you created so you can more easily track, because some people have trouble mentally sectioning that off from the larger tiles in order to get it. And several of the people that I taught, they actually really liked using that tool.
Oh, really?
Yes, they really liked using it because it made it much easier for them to figure out how many of each diatom and each color they needed to get.
I could see you using it for just the role play purpose of looking like you're holding up your little magnifying glass to the water to show the section you're working on, but interesting. I figured that was just for fun.
No, it's actually quite useful for many people. And that's most of the rules. It's actually a very simple game. It's a very elegant game. You can bring in additional judging rules to make it even more complicated if you want. And one thing is that once you set a diatom down on your board, which you have until it comes back to you. So this game could take forever if you had to wait for someone to place their diatoms before the next person person went, but you don't. You have until it's your turn again. But once they're down, you can't move them, which while being a nice standard game mechanic of like you have to commit, I also really like it because of the metaphor, because the metaphor is you're gluing these to a microscope slide. Once they're glued down, you can't move them. And so I think it actually works really well doing double duty there.
So we got this game, I think I picked it up at some point because it seemed like it was thematic. It was extremely pretty, but we actually had discussions. Is this a game we can talk about? Because this game, the metaphor is really about an obscure Victorian art form more than it is about diatoms. But we also thought, when else are we going to get the opportunity to have somebody on to tell us about diatoms? So we should just take this opportunity and talk about it anyway.
I'm much more liberal about what can go on. As long as it has science in it somewhere that we can talk about, I'm fine. And definitely, this is a science-light game. It is inspired by science. In fact, reading the designer diary, the tile laying component is a very early part of the game. That was sort of what the core was, but the diatom veneer, so the theming of the game, was actually a very late addition. The designer actually says how she was playing with the game, she had the tile laying down, it was fine. It was actually like collecting fractions because it originally was something to do about like math fluency due to some fellowship she'd gotten, but it really wasn't quite gelling. And then she took her child to the aquarium and they had some poster about diatoms. And unfortunately, the link to her picture was broken, so I couldn't see what the poster actually said. But apparently it talked about like these microscopic silicon-based algae that made these beautiful structures. And she just fixated on that. She started doing deep dives and researching, and she found this beautiful art form. And then apparently the rest of the game just fell together after that. Like it made perfect sense. She redid where you placed your little fraction things. Originally it was some sort of like bingo board into the actual diatom one. And that's fascinating to me because the metaphor feels like it goes so deep into this game in terms of how you actually play things. It's just fascinating to me that was actually the last addition to it.
Interesting.
But anyway, that's enough about the game per se. Now we're going to go to the actual science here. So, Laura, this is where you're going to need to help us because even though we both study microscopic things, we're both kind of bacteria people. Well, plants and bacteria. And my understanding is that a diatom is neither of those. So what is a diatom?
That is a question I get asked more than probably any other question in my career. And a diatom is neither a plant nor an animal. It is a protist. However, it is algae, which if you've ever looked at a phylogenetic tree of life, you'll notice there's the three main kingdoms. Algae actually spans all three, so it's not monophyletic, which is the term we use. But diatoms are a particular group of algae, and they all share the trait that they have a cell wall made of glass or silica.
Okay, so protists are single-celled eukaryotes. So they're not bacteria. They actually have complicated cells like we do that has a nucleus and all the organelles.
But that's okay. We're not going to hold that against them.
My understanding is that most eukaryotes are protists. This is another case where we are very biased by animals and plants and fungi because we can see them. But the vast majority of the tree of life of eukaryotes, of anything like us, is actually microscopic and we kind of ignore it. So you mentioned that they make a cell wall out of glass, out of silica. Why silica? It's like cellulose all over the place, chitin for fungi, peptidoglycan for bacteria, silica, glass. Why silica?
And it is their cell wall in the same way that like a plant cell wall has cellulose and pectin. They make their cell wall out of silicates.
Yeah. They do. So they don't make the silica itself, which if you think about it, that causes them to not have to use as much energy as it would to make an organic component. They pull it from the environment and then build the silica cell walls. So it's really readily available in the environment, which is an easy resource for them to grab. It's hard. It protects them from predators. It's opaline, and it has the ability for them to have pores, which allows for greater sunlight penetration for photosynthesis. So there's a lot of reasons why they have these silica cell walls.
So opaline, transparent?
Yep, they're opaline. They're not fully transparent. Of course, we don't really know because it's really hard to see a diatom and what it actually looks like. But the common belief is that if you know what an opal looks like, they're probably similar to that, where they kind of have this milky exterior that is radiant and then it has these beautiful colors to it. But we don't really know. They're so tiny. We can only see what we can see through the microscope, and they are see-through. So when you look at them alive, you see the chloroplast, you see all the organelles, you see this beautiful golden brown color when you look at them because you're not seeing the actual cell wall because it's mostly see-through and transparent.
So I have two questions. One question is, what does the name mean? What's the origin of the name diatom?
The name diatom comes from it being two parts. So it has two halves because diatoms are shaped like a box where they have a top half and a bottom half.
Okay, so probably Greek diatomos or something like that.
That sounds right.
The other question was, so they're photosynthetic, but they're not related to green plants. Is that right?
Correct. The algal group that is most closely related to plants would be green algae, but diatoms diatoms are not green algae. They are in their different kingdom.
Okay, different kingdom. So they're brown algae, right?
No, brown algae would be
No.
seaweeds. They're
Oh,
called golden brown algae is their common name.
okay. There's too many flavors and colors of algae, I guess. There's a reason they span, so many different branches. Golden brown algae.
Algae literally fits in every kingdom of the tree of life.
So algae is a little bit like when you say tree. Lots of different things make trees, right? An algae is just a way of being a living thing.
Yep, traditionally they're photosynthetic. I think that's the only characteristic that really pulls them all together.
And then you said that you're the collections manager at a diatom herbarium. Now, I'm familiar with plant herbariums where people take plant samples and they'll press them in paper and then they record like where they were recorded and when and such. And people use that to study the distributions of plants and such. I assume a diatom herbarium is similar, but can you explain to us like what exactly is a diatom herbarium and what's it used for?
The diatom herbarium operates very similar to the botany herbarium. We actually do have a botany herbarium in our institution as well, but we're not a part of it just because we are diatoms and just diatoms. That being said, we actually do have radiolarians and other things, but that's another topic for another day of just collections in general. But we have the largest collection of microscope slides in the entire world. So we have about 300,000 accessioned microscope slides, and we have accessioned means that it's officially cataloged into the collection. We have a ton of other microscope slides that have not been formally added to our database, given a catalog number, and I don't even know, probably another 100, 200,000 of those. And then we have the materials used to make the slides. So when we say materials, it's usually organic or digested material. And what we mean by that is when you go collect an algae sample. So algae is found everywhere, freshwater, saltwater, there's even terrestrial diatoms, and you collect the sample. And then to actually see them and to see the intricate structures of the cell wall, we have to soak them in acid to remove all the organic materials you find inside the cells because they're see-through. We can't see any of the structures without doing that. So then we preserve the materials, which is the digested material, our sample. And so we have that as well in the collection. And we probably have about 100 to 200,000 samples. And all of our slides and samples are from all over the world. And how they're used is that researchers will contact us if they're trying to study diatoms in a specific region to look at what was there 150 years ago to see how that compares to what's there. We have a lot of type slides. So if you get into taxonomy, in order to describe a species, you have to select a single specimen, which is a whole debate in the diatom world that I don't think we have time for today to use as the type specimen. So we have about 5,000 to 6,000 type slides. So if you're trying to describe a new species, you want to look back at the original type specimen to ensure that what you're describing has not already been described, and then also to look at comparisons of morphological features.
You have the diatom holotypes.
We have a lot of them.
We talked about the game holotype and dinosaur holotypes about a year ago. So what's your job as collections manager entail? Are you entering all these into the database, the ones that haven't been cataloged? Are you filling orders? Like, what do you do as your day job?
Is your collection digitized at all, or is it all?
Oh boy. So that word digitized. So yes, we are digitized. We do have an online database, which is it's been chaotic. So we had one database in a format that wasn't functional for many years. And then we moved to a different format in which we're having to rebuild it and get it back to where it's fully functional. A lot of our data is digitized in the sense of we have the metadata. We have a lot of other stuff too, such as like pictures of the specimens on the slides, which counts as digitized data. We actually have 3D microscope slides available online. So we have scans of our slides that you can operate similar to looking at a microscope, where you can focus in and out at different not different magnifications, but you can zoom in and out of that scan, which is at 400 times magnification. We also have a ton of other just stuff. We have a library, which we have digitized as well. We have records of that. And I have to manage all of that, organize it, maintain it. I have to care for the slides to make sure they're being properly stored and not getting damaged over time, repair slides if they're broken, if I can repair them. I have to send loans out if other scientists request that they want to see slides or take images of type materials to send to them. It's a lot. The better question is, what don't I do as the collection manager? Because on top of all of that, I also am a researcher myself and try to publish papers and work on research projects, various different topics with diatoms. I primarily focus on phylogenetics with them right now and taxonomy, but I also do ecological research as well. It's a lot.
Yeah, and so if you remember from our talk about holotype, I think we talked about phylogenetics and taxonomy there. Phylogenetics is the process of figuring out what is related to what. And then taxonomy is the process of like assigning names and species. And it sounds like trying to tell two species of diatoms apart is about as problematic as trying to tell two species of bacteria apart, which is to say that the natural world laughs at these artificial divisions that we humans have made upon them.
But they've got all the cool structures. So unlike bacteria, you've got a lot of morphology to look at, right?
Yes, they have numerous structures that are so immense that you wouldn't even be able to comprehend how many there are. And we're discovering new ones each day. When we look at them in a scanning electron microscope, we can actually see the very detailed structures of the cell wall at a level that we wouldn't be able to see under a light microscope. We can see the internal features because they have features on the external part, the internal part, on the sides. They're held together by silica bands, and even those can have features that can distinguish two species. Diatom taxonomy is really complicated. It's really hard to distinguish them. There's debates about what is a new species, what isn't. And then when you start adding molecular data, which is the DNA, we finally have started doing DNA work on diatoms. We're very behind on that compared to other organisms, but we work on that too. You would think that would add more clarity, but sometimes it creates more confusion. There's a ton of cryptic species, which are species that you can't tell apart morphologically by how they look, but they have different DNA sequences, which would indicate they're different species. And there's the ecological species concept of maybe these two diatoms have identical morphological features and identical DNA, but however, they only exist in completely different ecological environments. So are they two different species or are they the same? Diatoms just are the epitome of all of the issues with taxonomy and what is a species and what isn't.
So are they sexual or are they just asexual?
They do both. So they produce by binary fission, which I think is the primary mode of reproduction. So how that works is they have a top half and a bottom half. Those will split apart and the top half becomes the bottom half for the daughter cell. However, if you think of a box or a petri dish, one half is slightly smaller than the other. So as they produce, they go through this size shrinkage. So you'll have a great size variation, which also creates some difficulties when trying to identify them within the same species and can actually affect the morphological structures as well and how they look. And in order to get back to the original size, they have to undergo sexual reproduction where they produce an egg and a sperm, mate, and then create an oxospore that can pull silica from the environment to get back to the original size.
That's crazy. It actually vaguely reminds me of telomere degradation. Jason, explain what that is.
So telomeres are the tips of your chromosomes, which is what your DNA is bundled in. And because you can't copy all the way to the end of them, they tend to shrink over your lifespan. And there are complicated mechanisms to help take care of that when children are conceived and such. And that is not my area, so I don't know what those are. But this sounds amazing that basically every time they divide, they technically get a little smaller until they finally need to do sexual reproduction so they
start over.
So they can start over, so they can get back up to normal size. That's just fascinating.
That's so cool.
How long have diatoms been around? I mean, something this diverse and this widespread, I'm thinking hundreds of millions of years. Silica shells, those have got to be very well preserved in the fossil record. How long have they been around?
And I imagine from a species depiction, if you're using the silica shell, then you can be using the same species characteristics for fossilized silica shells from a long time ago versus the ones that are out right now.
Yeah, so the current belief for how old diatoms are is from the Cretaceous period. They're actually relatively new on the evolutionary scale. So dinosaurs came here first, and then diatoms showed up sometime afterwards towards the early Cretaceous period. And the reason we know this is because the silica cell wall preserves so well. They are heavy, so when diatoms die and they lose their buoyancy, they drop to the bottom of the water body that they are existing in. So whether that be a river, a stream, a bay, an ocean, and they lay on top of each other and create a structure similar to like a sediment core. So when you take cores, which is like a giant cylinder that you can put into the dirt, pull it up, and then you have different layers, you can actually see how the diatom community has changed over time, especially in water bodies that are more stagnant, such as lakes or bays. We've been able to date the sediment, and that's how we can figure out how old diatoms are. And the current findings is that it's the early Cretaceous period. But diatoms have changed rapidly since then. So what we see from the older diatoms, they look remarkably different than what we see in today's modern times.
So quick aside, early Cretaceous would be probably 100 to 150 million years ago. I had no idea we'd be referencing holotypes so many times on this episode. But changed drastically how? So listeners, if you're not driving, find a place to do a quick pause, do a search for diatom art or diatom diversity, and just spend a few minutes going down the Google rabbit hole of just how varied and beautiful these little things are. When they're mounted, they're glass shells. You hit them with a light, they turn all these beautiful rainbow colors. I look at those like we have long skinny ones, we've got round ones, we've got ovals, stars, these weird triangle things. If you're saying they were very different than what they were, what were they?
I'm trying to think of how to describe it without showing a picture, and they have strange structures. They almost look alien. Some of them have what looks like antenna, weird spines. The areolae layer, the pores in the cells can look really strange compared to what we see today. And in my mind, when I think of a normal diatom of modern time, I have the background to know what that is, but it's hard for me to describe that without using pictures of what the shapes and everything look like. But even the shapes of them back then are different than what we see to now. Let me start by saying there's two main classes of diatom shapes, which is centric and pinnate. Centric are more circular and pinnate are more boxy. So centric diatoms are believed to have happened before pinnate. So that's why you'll see more centric diatoms when you look back in the course.
I'm just thinking how every time life hits on something new, it tends to do it weird at first, which obviously is our own personal bias. But I'm thinking of if any of you have ever seen pictures of animals recovered from the Cambrian explosion, which was when animal life first really diversified in the oceans about 500 million years ago, I think. There's some weird critters in that we look at those today as like that was evolution experimenting because it hadn't figured things out yet.
So we had the diatom explosion of the early Cretaceous where they were experimenting with form and shape. We'll definitely try to find some pictures that we can put up with these or point people to things on our show notes so we can say like, this is a current diatom, this is an early diatom, so that they can see for themselves how they have changed over 100 million years.
I definitely recommend doing that. If you're ever looking for a good rabbit hole to get stuck in, diatoms is a great one for that.
Do you have any diatom channels you can recommend on social media?
I don't know if there's diatom channels per se. There is the DONA, which is the Diatoms of North America. We have a webinar every other Tuesday at 12 Eastern time, and they save all of those online and publish them on YouTube, so you can watch all of the previous webinars. Some of them are a little bit more friendly to the novice, and some of them are more geared and tailored towards researchers, but there's a large variety of the different webinars, and those are fun to look over. There's also Jeffrey Stone. I think he has an Instagram or some sort of social media account where he does SEM work, which is the scanning electron microscope, and I'll often show diatoms and talk about them. I think they actually have their own podcast. I'll have to do more research into that and give you the name of what they do.
Oh, that would be great. So I manage the social media for as it is for the podcast, and I know that there would be a hunger for diatoms on Instagram. There's a whole subdivision of Instagram people just taking pretty pictures of things in nature and micrographs and close-ups of plants and everything. And I think diatoms would hit big.
Unfortunately, I think our biggest contender for that we lost a few years ago. So if you do any search for diatom art, you will eventually run across Klaus Kemp, who until a few years ago was, as I understand it, at least the most famous living diatom artist, possibly the only professional living diatom artist, but he passed away in 2022. But he actually spent years perfecting a glue recipe that would take days to dry so that he could position his diatoms properly, because there's hundreds of these things and it takes a very long time. He built custom microscope rigs. People like pick them up and move them around. There's a video you can look up called The Diatomist. I'll link it in the show notes. It's actually well worth a watch. It's only about 10 or 15 minutes long. Beautiful. Highly recommended.
So if you want to manipulate a diatom on a sled, are you using like a super pulling like a fine glass rod into like a little micro point or like a fine needle? How do you move one diatom?
So there's a lot of people who've tried various ways to do that. I know actually a decent amount about the original diatom arrangers from the Victorian time period. We have a lot of slides by Moeller, who was the original or most well-known diatom arranger from that time period. And so I've done a lot of research because there's some of the more precious items in our collection and they're fun to show to the public, or if we have any events. I've done a little bit of research into his life. And one of the things I've noted most is that the original arrangers all kept their secrets with them and how they did it. So fortunately, we don't know what they did to create these beautiful arrangements, especially Henry Dalton, who used butterfly skulls, parts of bugs, and created true artwork. He has chickens, flowers, they're just insanely gorgeous. I also recommend searching Henry Dalton arranged slides. They're gorgeous. And there are also some diatoms on there that he dyed to add color to them. But Klaus Kemp was the most modern one, and he has records of how he did it. But how you move diatoms, we have to still do that if we want to do SEM work and look at a certain position of a diatom, because they look a lot different when they're sitting on their side versus when you see them from what we call valve view, which is the forward view. We used glass pipettes that we stretch under a flame, so they're super tiny. We tried using that to suck them up and move them around. It's really complicated, really hard, very tedious, exhausting, stressful work.
That sounds like the voice of experience.
Yeah. Trying to make monocultures for diatoms is one of the most frustrating things I've ever done in my life, because you have to isolate one cell and move it because they do reproduce by binary fission. And there's not enough DNA in most cells to get quality DNA sequences. So you have to isolate one cell, move it into a petri dish with medium, and then you just pray that it grows. It often doesn't and just dies. But that isolating one from a live slide culture is just frustrating. But we actually have a student. Her name is Sylvia Lepic, and she really found these arrangements arranged slides beautiful and loving. So she started doing that herself. And she uses a eyelash glued to a wooden rod that she will look at the microscope under 100x magnification and manipulate diatoms that way. I forgot to ask her what kind of glue she uses. I know she used glycerin plus some lab grade gelatin, as she tried that as well. But she's actually been really good and has made some gorgeous arrangements just out of fun.
That's really cool. I'm thinking about these other times where you see the science and art kind of coming together. Like I'm thinking about the glass flowers that are on display. I think it's at Harvard because they're so important for identification, but they don't flower all the time. So if you really want to be able to teach people how to do the identifications, you have to have the flowers. And in this case, the sort of art made with the diatoms, I don't know. I mean, like if you could make a diatom arrangement that would be a combination of art and science, would you try to arrange like a phylogenetic tree? Like what would you make as your arrangement of diatoms?
There's actually an example of what I really enjoy about the arranged diatoms. So as I mentioned earlier, that we have to see certain features with the different views that you see them in versus girdle versus valve view, is there was Schultz, who was a original member of the Academy of Optical Sciences, who made arranged slides, and he would take valves and put them in these different orientations so that way they could be used for research and to really learn the morphology of the diatom. So I would enjoy doing something like that. There's also Moeller was quite famous for using microphotography, where they would take a picture with the names written of the diatoms and somehow shrink it down where they could glue it onto a microscope slide. And they would place individual valves into the circles above these names. So when you go to look at this microscope slide from the 1800s, you see a diatom and under it is the name written down.
Amazing.
And I think that is amazing because back then he paired up with Rabenhorst and they actually did do art compared with science. It was an amazing learning tool. They were able to help young taxonomists start to learn the name so they have access to the diatom right next to the name.
Oh, it's a lovely collaboration between science and art. We don't get enough of those, but they're wonderful when they happen. In all of this, Laura, do you have a favorite diatom? I mean, you have access to the world's largest collection. Surely you must be able to pick a favorite out of all that.
And I actually wanted to ask a slightly different question that's on the same theme. I see that you have named you've contributed several species. So is one of those your favorite?
So right now, my favorite diatom, it does change fairly often depending on what I'm working on, is I recently published a paper where I described a few new species of Pinularia. And one of them was able to name after my former research advisor, Dr. Kalina Manoloff. So that's very special to me because I was able to do something special for her and give her back a little bit of how much she's poured into my life and my professional career.
That's very sweet. What is the name of that species?
It's actually not on Diatoms North America because I need to make a page for it still because the paper was just published recently. So I couldn't have made a page without the name being officially published.
Gotcha.
But it's Pinularia Manoloave.
Okay. Well, let us know. We'll point people to it when it's out, okay? We can at least put it on our Discord.
Going back a little bit, you talked about how diatoms stick around. And when I was researching, I came across something that I've run into previously, which is diatomaceous earth, which is apparently dirt made from diatoms. Can you explain to me what this stuff is?
Yes. So it's deposits of diatoms that if there was a body of water that had an abundance of diatoms, as it dried up over time, all of the silica would sink to the bottom. And then you just have this massive deposit of pure silica dust. And it appears white and chalky. And it's just crushed up our full brustles, which is what we call diatom cells. And it has a lot of applications. It's used a lot for commercial use. It's a very valuable resource. And it's also beautiful to look at because it's just a whole bunch of diatoms. And I know of a few things they use it for is like a dynamite stabilizer. It's used for filtration to make beer and wine and fish tanks. They use it as an abrasive, so for fine sanding because it is slightly abrasive. It's found in some toothpaste because it is a gentle abrasive.
We use it for pest control in organic farming. It's the insects don't want to walk over it or something. I don't know.
What it does is it gets into the skeleton of the bugs and breaks it apart. So it kind of is like a whole bunch of tiny glasses attacking the bug.
Even
Tiny
worse.
glass knives being shoved into the bug's exoskeleton. Got it.
But it's organic and won't harm us as humans.
Like eating glass isn't bad for humans, or it's too small to hurt us.
You can buy food grade diatomaceous earth, and there is a difference between food grade and non-food grade where it's fine to ingest in small quantities. It's not fine to breathe in, and it actually can cause lung damage if you do breathe in too much of it.
Ew,
because it is just
interesting.
tiny shards of glass.
Okay, so okay to eat, not okay to breathe.
Exactly.
Got it. Also, part of me just chased at the idea of a silicon-based substance being called organic.
That's a different episode completely.
That depends on the definition of organic.
Yes, it does.
Yes, I know. So one of the facts I ran across when researching for this episode is that apparently the Amazon rainforest gets fertilized by diatom dust from Africa. That like gets blown across from the Sahara from where there used to be a bunch of lakes and they've all dried up now. Can you confirm that?
I do know about that. I don't know a ton about it. I know very little bit, but if you do go and you collect some of the sediment off of the top of the Sahara Desert, you'll find diatoms in it. And diatoms are remarkable organisms and can withstand years of desiccation. So there are, I believe, confirmed instances where they've been able to rehydrate diatoms from the Sahara Desert, which would have allowed for that repopulation if it gets blown in the wind or gets carried up in the clouds and then dropped on the Amazon rainforest. The whole idea of diatom dispersal and how it travels around is a very complicated topic.
Okay, Brian, diatoms have just surpassed tardigrades in terms of my favorite cute microscopic organism.
Okay.
They don't look like bears. The tardigrades are adorable.
Yeah. So like what are the okay, so I was gonna ask about the DNA thing, but then I figured it might be like a touchy subject. I'm also thinking about how everybody's taxonomy got rewritten by DNA. For bacteria, it's mostly settled down. The fungi people are going through it now. But for the diatoms, it seems like it's even worse because the problem now is that you've got a morphological species concept that you've been able to apply back through time, but you can't do that with the extinct ones. So what you're going to have is two parallel species definitions for the same groups of organisms. That's going to make things complicated.
I would say that is a touchy topic, and there is two different sides or parties that believe certain things. So diatoms are used as bioindicators, which is one of the biggest applications for them in environmental research. So a bioindicator is an organism that you can, based on its presence or absence in the environment, can give you ideas about how healthy or clean or unhealthy the environment is. So diatoms are used as bioindicators, and that's all been based on morphology for the past however long we've been working with them. But now there's this metabarcoding and eDNA that's coming up, and you can use that to make water quality research using diatoms. However, you lose that very important connection between how they look and the DNA sequence, because eDNA does not allow that connection to morphology unless you have a reference library that was done through monocultures to be able to compare the sequences.
All right. And so eDNA just means environmental DNA. It's DNA you get from like taking a water sample or a soil sample instead of taking it from a specific organism.
So there is this debate on, do we need to connect the data? Do we not need to connect the data? And it can get a bit dicey between people on this debate. And generally the diatom community is very friendly and cohesive. However, debates do happen in any scientific community. So this is definitely one of them that happens fairly frequently. And there are people who just do diatom DNA and don't really know anything about morphology. So it's a strange and complicated topic.
I feel like if the diatomologist and the artist can get together, then the molecular and the morphologist should be able to find common ground as well, right?
I would hope so. And I definitely am on the party of, or we need to figure out how to connect the morphology with the molecular data. And there are a lot of people that are working towards that and stand in that middle ground. So there is progress moving forward.
Diatoms united.
So you mentioned that they're indicator species, and you mentioned previously that they fix a lot of oxygen for the world. So what else do they do in ecosystems? What other roles are they playing?
So diatoms are the primary producers as almost all aquatic ecosystems. If you think back to 5th grade science, when you learned about primary producer and then the grazers, and then you have the predators. So everything eats everything, and the energy flow through an ecosystem all starts with plants. Well, diatoms and other photosynthetic organisms play that role in aquatic ecosystems. So they're very important because without them, you wouldn't have the energy flow through the ecosystem.
So they're the basis. They will capture the sunlight and it flows through. And because they're largely microscopic and just kind of like the brownish slime that we see on rocks and tree stumps and stuff, we just ignore them. But it's actually a really important component for how energy flows through the environment.
Yes. And diatoms in particular are interesting because they have lipid droplets, which is oil in them. There's this whole idea to use them to milk for oil. But again, that's another topic that requires a lot of conversation.
Did you say milk? Are we milking diatoms?
I did say milk. So when they get stressed, they produce lipid droplets, and that's oil. And so the idea was to get a whole bunch of diatoms in a pool, stress them out, and then milk the oil out of it.
Oh, wow. Is there a society for like protection of diatoms? This sounds cruel.
No. The problem is it takes so much energy to get to that point where you actually get a usable amount of oil, is it's not really any benefit. It's actually worse than the regular oil that we use now.
Huh.
But they've been referred to as the hamburger of the ocean because they have those oil droplets as they're very nutrient dense. So they're beneficial for organisms to eat them, especially the macroinvertebrates or the small fish that eat them.
Do the lipids contribute to buoyancy and keep them up in the photic zone, or is that just am I just making a connection to something that isn't actually there?
I have to go back to my plant
Okay.
cell anatomy, but I think it's primarily to use for storage because they do it when they're stressed, when the environment. So I think the storage are a way to keep them alive under stress.
Okay.
So, what is the weirdest environment that you have a sample from?
So I don't personally have any. I mean, the collection, I think we have some from some hot springs in Japan. My boss here, the curator, Dr. Marina Potapova, has a lot of Arctic diatoms in her collection and some really harsh environments that they also look a lot different than what you see, such as like temperate areas, Philadelphia region. But I would say the hot springs probably is one of the most interesting samples we have because you would think it's quite strange that diatoms can survive really intense temperatures, but they can.
There's something really lovely about the thin brown slime layer. And then when you look at it under a microscope, it's this incredibly gorgeous complex.
Yeah, I wanted to try to do our own diatom isolations in preparation for this episode, but it's a very busy time with grant proposals and such, and so we were not able to do that. Although we have plenty of ponds around campus where we could go get some, and I'm sure Brian has some hydrochloric acid or something that we could boil them in to clear them out. There are videos on YouTube that will show you how to do this. Just be very careful doing it at home because you do have to like boil hydrochloric acid or stuff like that in order to clear them out.
Hydrogen peroxide also works really well.
Oh,
If
that
it's
I
not,
actually
if
do
you don't have a
have.
ton of organic material in it, it's a softer treatment. So any diatom that's lightly silicified, we have to use that for. But it works pretty well, like 70% hydrogen peroxide, even like 40% hydrogen peroxide. I don't think it's the kind you can just buy from a drugstore. I haven't tried that before, but it works. And you do really need a fume hood to do this. I do recommend not doing this without a fume hood.
Well, then, Jason, actually, yes, I have both of those things. So bring me some pond slime and we can do it later, okay?
Okay, so we've talked a lot about the morphology, and these things are beautiful. Now, there's a bunch of shapes in the game. It's conveniently, they tie the shape to the number of color patches you've gotten. So if you get one patch, it's a circle, two, it's a little oval because it has two sides, three is a triangle, four is a square, five is a star. Looking online, I saw a bunch of much weirder ones where they're like bulbous or where they're like long and thin. What is the weirdest diatom shape out there that you've seen? Like, are any of these things like weird fractal shapes or anything like that?
The weirdest one I always think of is Campylodiscus, which looks just like a Pringles chip.
Okay, microscopic glass potato chip. Got it.
Yep.
It's probably very crunchy too.
I'm sure it's very crunchy. I'm sure your teeth would not like it.
There's also some too, like Entanemus, which look like a figure eight, but they're also kind of twisted.
Like a Mobius strip?
I don't know what a Mobius strip is.
It's like you could take a piece of paper and twist it on itself so it technically only has one edge.
I think it's similar. So they look twisted, and they have pictures of them on Dona, which is the diatoms in North America.
There will be many, many photos linked in our show notes, people, so you should check them out. Okay, so we should start wrapping this up, which comes to our nitpick corner, Brian's favorite part of the show.
Oh, I don't know, man. I don't know what to talk about. I mean, yes, you get diatoms out of water. Like, that's a thing. Yes, people used to arrange them on slides to make pretty murals. That's a thing.
I don't think there's much we can science nitpick in this. Like, there's
No.
not enough science in it. It's a very science-like game. I think we can nitpick the one issue. The game pieces are beautiful and
Yeah.
they're all foiled, but the problem is the foil actually makes it hard to see the color sometimes.
A little bit.
So when it comes scoring time, I was lifting up
Yeah.
my scoreboard and I was kind of like tilting it to try to get the light right. It's like, is that red? Is that purple?
The patterns of the pores, you said those are the
Areola, yeah.
areolae. Those are different on each shape. So you can use, they're colorblind friendly in that regard. So they always have a pattern there too. It's not just the color, but you're right. Like the yellow and the green, really hard to tell apart if you're in the wrong kind of light.
But that's about it. Like I said, it's a very elegant game, very well put together, very high quality, and I find it very fun. I don't know. I've toyed with the idea of going through the single player mode. So single player mode involves a bunch of cards where there's challenges and there's rules so that you can't just like play it forever until you get it. You only have so many spare diatoms you can pick up that don't fit the patterns you're trying to do. And they're actually really quite fun. And you start actually on the front of the board, which has all the set little pieces I mentioned where you can only put a circle or a star or whatever. But the second half, you flip the board over where it doesn't have that and it's freeform arrangement. And so they have some of these where you're making arrangements that look like a tiny soul solar system, or ones that look like a face or a tree or other things like that, where you're putting these together.
Jason is holding up cards of the different shapes. So for those of you who, Jason's forgotten that this is an audio podcast.
No, I'm trying to get your reactions. Like, I know our audience can't see, but you can at least react.
Audience, I apologize for Jason.
All right. I mean, that's really it. There's, again, elegant game, very well done. So on to grades. And I'm going to say this is, I think Turing Machine was the one where I gave an undefined science grade.
I think what we need is an art historian to really grade this. I think that this is the wrong class.
Okay. I will say the science grade is undefined, but I definitely want to give it points for bringing attention to these beautiful creatures. And okay, it does actually do some things right. It gets the shininess of them right. Well, it gets the shapes right. It gets the variability there. I don't want to give it a grade because that implies a wrong standard. But I want to say that they managed to make the game and the science mesh beautifully, and I really appreciate that.
We wouldn't have had a chance to talk about diatoms otherwise. So, like I said, I'm not going to grade it either, but I'm glad it's out there. I'm glad it exists. Not for science. We can give it a fun grade. Well, actually, Laura, what do you think? You haven't had a chance to play, but it sounds like you'd be more informed on this than we would be. What do you think?
I'm just happy there is a board game out there about diatoms because I'm just happy that diatoms are getting promoted in any way possible. So I'm very thankful for that. And I have seen it before. It does look like a beautiful board game. And honestly, I think it kind of gets the heart of whole diatom arranging and the really complicated nature of the rules almost as a perfect analogy for how complicated and complex diatoms are. So I really like the game. I'm going to buy a copy eventually.
Awesome. Let's see. Jason, I want to hear your fun grade first because you really like this game. In fact, I bought the copy, but you have the game because you like it so much more than I do.
I'm going to give it an A. I keep using the word elegant. I love things that are elegant, that have relatively simple rules and yet have a lot of strategic depth to it, which I think this game satisfies. And like I said, it's one of the few games that we've played that has a solo mode where I've actually played the solo mode and enjoyed it, and I'm considering doing more. There may be like 30 days of diatoms on our Instagram feed at some point.
Jason may not want this shared, but one of his hobbies is stained glass. So I think this is merging his love of highly analytical optimizing games with his desire to create beautifully arranged mosaics.
Oh, yes. The fact that these are actually made of glass is part of that. Taking something from silica, making beautiful arrangements. And so, yes, there is a little bit of synergy. It's like, oh, this is that same thing, but on a microscopic scale. I don't think we've mentioned it. So we talk about all these diatom arrangements on microscope slides. So like a full beautiful mosaic of like 100 or 200 little diatoms in these beautiful patterns is about the size of a period. So they really can only be appreciated under the microscope or as prints.
I think it's even smaller than that, depending on the size of the diatom, because the largest diatom is only 2 millimeters wide, and then they get much smaller than that.
Wait, they get up to 2 millimeters?
Mm-hmm.
That's like naked eye visible. Where is this diatom?
It's over there. Jason, look behind you.
I'm trying to remember what it's called. It starts with an E. I think it's Entomodiscus, but I don't know exactly where it's found.
Entomodiscus. Insect circle?
Is that an insect associated? Is that a thing?
I know it's a big circular diatom. It says it's found in the temperate zones of the world's ocean.
Oh, okay. For my fun grade, I guess I have never gotten out diatoms to play except for you, and then you took the game. So, and then
you gave me the game. I did not take it.
I did. I have actually officially given you the game. I have been like, Jason, you should keep this game. You really like this game. I don't know. We could play it a couple more times. I don't really want to ding it. I'll just say B. It's fun. I enjoyed it. It's a little complicated. It's kind of 2 games in one. There's the selection, and then arranging the mosaics is very complicated. And you know how I feel about over optimizing.
I enjoyed it, and I actually got to play it with my 9-year-old, and she was also enjoying helping me make the arrangement and stuff. So I consider that a win.
Absolutely. Laura, I just wanted to ask, do you have a favorite game?
I actually love classic Monopoly just because I get to wreak havoc or chaos when I play it.
Oh, really? Do you play with house rules or do you play by rules?
Well, when I do play with my brother, primarily, it's rules, and we get very competitive with each other. And we actually have a long history of using my cousins as pawns to beat each other.
Monopoly, bringing families together for however long Monopoly's been around.
Does that mean you're using your cousins as like shell corporations in the game?
Yep. I have no shame over it either.
That's fantastic.
All right, well, we'll wrap it up there. Thank you, Laura, very much for coming on and telling us about these beautiful microscopic organisms. Is there any place you want people to check you out? Do you have like a social media or something else to follow?
They can check out Diatoms North America, which is a wonderful resource for them to use. For Diatoms, I am a part of that. And then if they want to ever come see Diatoms, we do have a very small exhibit at the Academy of Natural Sciences in Philadelphia, which is the oldest natural history museum in the entire country.
Well, next time I'm in Philadelphia, I'll put it on the to-do list for sure. Sure.
Well, now you have something for your travel plans, listeners. So with that, we will sign off. Thank you very much. Have a great month and good games.
And have fun playing dice with the universe. See ya.
This has been the Gaming with Science podcast, copyright 2026. Listeners are free to reuse this recording for any non-commercial purpose as long as credit is given to Gaming with Science. This podcast is produced with support from the University of Georgia. All opinions are those of the hosts and do not imply endorsement by the sponsors. If you wish to purchase any of the games we talked about, we encourage you to do so through your friendly local game store. Thank you and have fun playing dice with the universe.